Okay, so as Saurabh mentioned, sorry, maybe I'm not going to be able to use this. Use this one. Sorry about that. Okay. So as Saurabh mentioned, we've worked on a number of problems in my group over the years, but a problem that we keep coming back to is this one addressed by the FastCons method that Saurabh mentioned, and here's a figure from that paper, which is now 13 years old. So for those of you who don't know, this is a method that measures evolutionary conservation based on alignments from multiple mammalian or non-mammalian vertebrate genomes. And it's based on a statistical phylogenetic model combined with a hidden Markov model we call the combination of phylo-HMM. And it can be used to identify not only coding sequences that are under strong evolutionary conservation, but it also very nicely detects many unannotated sequences in mammalian genomes that are also under evolutionary conservation. And as such, it is a tool for discovering elements that might not have shown up using other high-throughput experimental technologies. So this turned out to be a very useful tool, and there are many other tools that address a similar problem using similar techniques. But they do have some important limitations. One limitation is that they require getting the alignments right, which, as many of you know, is non-trivial. They require properly identifying orthologous sequences across long evolutionary distances. And these methods also sort of make an assumption that the elements that are functional in the human genome are also functional in the genomes that are being aligned to the human genome. So in the case of elements that have been around for a long period of evolutionary time, like many protein coding genes, they work very well. But for functional elements that come and go over evolutionary time, that are gained and lost or experience turnover, these methods can fail because they're essentially averaging the evolutionary pressure over tens, possibly hundreds of millions of years. And so they don't work in the case where elements are under differential evolutionary pressure over long time scales. So for several years, we've been thinking about ways of improving these methods by a variety of different techniques and taking advantage of some of the new data that have arisen in the last 10 years or so. And part of this has involved taking advantage of all of the high-throughput epigenomic data generated by the Roadmap project and ENCODE and many other projects, using those epigenomic signatures to help guide these evolutionary methods toward regions of particular interest. But in addition, we've been thinking about how to measure natural selection on much shorter timescales, taking advantage of patterns of genetic variation within human populations. And I'm going to tell you about some methods that we've introduced over the past few years that try to take advantage of both of these properties, epigenomic data and evolution on much shorter timescales. Before starting, I wanted to say a few words about both the limitations and the strengths of evolution-based methods, because I think we live in a different world than we did in 2005 when we first started making use of evolutionary pressure to identify functional elements. But as I'll argue, these evolutionary methods remain relevant and useful, even when compared with CRISPR screens and massively parallel reporter assays and other newly emerging high-throughput experimental methods for identifying potential functional elements. So first, let me talk about a few of the limitations. Obviously, natural selection is not the same thing as any type of molecular function, and neither of those is the same as disease relevance. So we have to keep that in mind when we use these evolutionary methods to identify, let's say, variants that might be associated with diseases. One component of that of particular importance is time scale. So the time scales that we're measuring, even if we use population genomic data, are on the order of hundreds of generations or thousands of years, whereas some of our interest in biomedicine is usually limited to much more recent time scales. The other important limitation of these methods is they require some sort of a realistic and interpretable model of evolution. And the reason is that we can't really observe any of the details of the evolutionary processes. We only see sort of noisy outcomes from that process. And so we need some sort of a statistical model to try to say something about evolutionary pressure, in particular selection pressure, over many generations based on our observations of, say, genetic polymorphism or divergence between species. On the other hand, these methods, I think, have some important strengths that make us continue to think about how to make use of them. I think one particularly important strength is the idea that these methods naturally are integrating over different cell types, different conditions, and different developmental stages in a way that you don't get if you're using something like a massively parallel reporter assay, where you typically have to choose a particular cell type and condition for your experiment. So these evolutionary methods are naturally thinking about all of the different relevant cell types and stages and conditions that contribute to evolutionary fitness. That, I think, gives them an important advantage over some of these newly emerging experimental techniques. Similarly, they're measuring fitness of real organisms in their natural environments, not in some sort of artificial laboratory experiment. Many elements that show no effect, for example, in a reporter assay, may have some effect in a natural environment under some set of conditions. It's just that we haven't tested those conditions properly in the reporter assay. And the other thing that I think is important about these methods is they're exquisitely sensitive to small effect sizes. So over many generations of evolution, you get this amplifying effect of effect size, so that effect sizes much less than 1% can still be very easily detectable in terms of patterns of genetic variation. Whereas if you're doing an experiment like a CRISPR screen or something, you probably will not be able to detect an effect size anything close to 1%. So if the assumption holds that evolutionary pressures have been similar over long evolutionary time periods, then we can potentially detect even very, very subtle effects that variants have on fitness. Okay, now I'm very briefly going to introduce a method that we call INSIGHT that we published now 5 years ago, but that forms the basis of several other methods that I'm going to tell you about. I won't be able to go into any of the details, but this is published work, and I'll show the references at the bottom of the screen as I go along. So the basic idea of INSIGHT was to take a collection of pre-annotated elements across the genome, such as transcription factor binding sites identified by ChIP-seq data, and then to systematically compare those elements, shown in yellow here, with nearby putatively neutrally evolving sequences, shown in gray, and to compare them in terms of their patterns of both genetic polymorphism within a panel of individuals, in this case a panel of human genomes, and their patterns of divergence across a phylogeny with closely related relatives, in this case non-human primates. And by picking up on systematic differences between the elements and these flanking neutral regions, we can develop a robust, unbiased statistical estimator for the fraction of nucleotides that are under selection, allowing for several confounding factors, such as variation across the genome in mutation rate and coalescence time. And so we showed through a series of papers based on both simulated and real data that we could obtain useful measures of the effects of natural selection on collections of dispersed elements across the genome, like transcription factor binding sites. And we were able to show that many transcription factor binding sites are under fairly substantial selection pressure, not as substantial as protein coding genes, but still quite strong effects. On average, we observed about 30% of nucleotides in transcription factor binding sites identified by ChIP-seq peaks were subject to detectable natural selection. So here's a slide just sort of showing the probabilistic model that underlies this method. I'm not going to walk through it in any detail, but I just want to mention that it has three components. There's a simple polymorphism model. There's a model for evolutionary divergence over medium timescales. So in this case, we use a Jukes cancer model that describes evolutionary divergence since the human chimp ancestor, leading to the human population. And then there's a statistical phylogenetic model that basically serves as a way of pulling in information from outgroup sequences. And its real importance in this method is just to give us probabilistic information about ancestral alleles. So it effectively probabilistically polarizes the ancestral allele and gives us a better ability to interpret the human data as derived or ancestral alleles. There's a probabilistic model that describes this process. Basically, it's a model that describes the evolutionary process at every site. In the sites of interest, the target sites, such as the transcription factor binding sites, it's a mixture model of selection and neutrality. And then in the flanking regions, we assume that complete neutrality. We assume these regions are completely neutrally evolving. Now, if some of them are experiencing some selection, as is inevitable, that'll just make our estimates conservative for the fraction of nucleotides in the target sites that are under natural selection. All right, and this method was developed by two postdocs in the lab, Ilan Grunau and Leo Arbiza, and published in 2013. Okay. So, one more thing about that. So the key quantity that I want to emphasize in this talk that we can pull out of this INSIGHT model is this parameter we call rho, which represents the fraction of nucleotides in the target sites that are under natural selection. So if we get an estimate of rho equals 0.3, that tells us 30% of nucleotides in the, let's say, transcription factor binding sites that we're analyzing appear to be under natural selection. According to the difference in the patterns of polymorphism and divergence within those sites as compared with the flanking neutral sites. And what really drives this method, there are several factors that come into play in estimating the fraction of nucleotides under selection, but what really drives the method is the depletion of both polymorphisms and fixed differences in the elements. So for example, if we see a depletion in transcription factor binding sites of polymorphisms, of SNPs, relative to the flanking neutral sites, and a depletion of fixed differences with respect to, say, the chimp genome within those sites as compared with the flanking neutral sites, then that's going to drive our estimate of what fraction of nucleotides in those target elements are under natural selection. Okay. The method also lets us estimate some other quantities that I'm not going to emphasize in this talk, but I'll just mention them briefly. It gives us a way to estimate, to quantify the effects of positive selection and weak negative selection, selection that's strong enough to hold polymorphisms to low derived allele frequencies, but not strong enough to prevent them from being polymorphic at all. And in our original papers, we make some use of these methods, but in today's talk, I'm just going to focus on this parameter rho, the fraction of nucleotides under selection. Okay, so now I'm going to move on to sort of the second idea of the talk, and that is turning these estimates of rho, fractions of nucleotides under selection, turning them around and using them to measure something slightly different. So this was an idea that we had after finishing up the INSIGHT project, and you can think of it essentially as a way of hijacking this probabilistic model for another purpose. So the idea is that instead of having a collection of annotated elements across the genome, like annotated transcription factor binding sites, we're now going to assume we have nothing annotated in the genome. We just have a collection of high-throughput epigenomic data sets. So things like RNA-seq, DNA-seq, histone modification, ChIP-seq data, and so on. And then we're going to cluster sites in the genome by their joint patterns of these epigenomic features. So for example, one cluster might have, a high RNA-seq signal and a low DNA-seq signal, and histone modifications that are, indicative of promoters or something. And there would be many other combinations of features on the order of several hundred combinations of features that would be that would distinctly identify collections of sites across the genome. We call these the epigenomic fingerprints of those regions. So we might have, say, 500 or 600 different epigenomic fingerprints. Each of those epigenomic fingerprints would identify a collection of nucleotide sites across the genome. And now I've just shown you a framework for estimating the influence of natural selection on a dispersed collection of sites, very much like these dispersed collections that are assigned to these clusters. So the next step of this method is we simply feed each of these clusters of dispersed nucleotides into the INSIGHT algorithm and obtain an estimate of this quantity that I've called rho, the fraction of nucleotides under selection. Now, an alternative way of thinking about the fraction of nucleotides under selection for some arbitrary dispersed collection of nucleotides is that if I were to randomly mutate a nucleotide in that collection of sites, then that quantity rho also describes the probability that that mutation would have some effect on fitness, right? Fractions of nucleotides under selection are sort of the mirror image of probabilities of mutations having fitness consequences. So we're now going to take this quantity rho, the fraction of nucleotides under selection, and interpret it as what we call a FitCon score, a score indicating the probability that a new mutation in a collection of sites across the genome will have some impact on fitness. And now we can map those scores back to the genome. So for a particular cluster, one of the 600 clusters, we get an estimate of rho of, say, 0.15. Now, every nucleotide in the genome that belongs to that cluster, we assign a FitCon score of 0.15. Okay, so when you do that, something kind of interesting happens. You end up with a plot, shown here in the middle, that looks a lot like a conservation score, right? It's elevated in the exons of this MIR2 gene. It's kind of somewhat elevated, but not as elevated in the untranslated regions. We see a spike at the promoter, and occasionally we see things that look like regulatory elements, either near or far from the sequences. Now, keep in mind, even though this looks qualitatively like evolutionary conservation scores, it's actually measuring something quite different. When I look at the score for a particular region, such as this one here, what I'm actually measuring is the probability of fitness consequences across the entire genome of all sites that share this site's epigenomic fingerprint, right? It's a little different from actually getting all of that information locally. But that difference allows me to pool data effectively across the genome and measure things on much more recent evolutionary timescales. I'm now able to measure evolutionary pressure, fitness consequences, on the evolutionary timescale associated with human populations and the divergence between humans and chimps about 5 million years ago, rather than on the timescale of 80 to 100 million years that I have in the comparative genomic analyses. And as a consequence, you see sometimes you pick up things that don't show up well from evolutionary conservation scores. So look at this putative enhancer out here upstream of the MIR2 gene. You can see that it shows up with an elevated FitCon score reflecting a strong DNA-seq peak and chromatin marks indicative of enhancers, but it doesn't show up well with philo-P scores or FASCON scores. And that may very well, in this case, represent a newly emerged enhancer, an enhancer that has emerged more recently enough that that we're not well powered to detect evolutionary patterns from long evolutionary distances captured in these comparative genomic alignments. Okay. All right. So this was the next chapter in this sort of saga, this series of methods. And we published this method in 2015, but this method also has some important limitations. And the main limitation in this method is that this clustering scheme that we're using scales very poorly. So essentially what we were doing in this method was we were pre-annotating every site in the genome as having one of a discrete collection of levels for each covariate of interest. So for example, low, medium, and high RNA-seq, or low, medium, and high DNA-seq. And then we would consider the Cartesian product, all possible combinations of those labels in order to get at these joint epigenomic fingerprints. But the problem is the number of possible combinations blows up as the number of covariates gets large, right? So this method doesn't scale very well. And the fundamental reason it doesn't scale very well is we're doing the clustering in a way that is completely agnostic to the downstream evolutionary analysis. We're pre-identifying these epigenomic clusters rather than having the evolutionary patterns guide the clustering analysis, right? And so now I'm going to show you some ways of getting around that limitation that allow us to consider much larger numbers of epigenomic marks in these kinds of analyses. Okay. I'm going to show you very quickly two methods that I don't have time to talk about in detail, but I want you to know about them. The first is called LINSIGHT. This was developed by a postdoc in the lab, very talented postdoc, Yifei Huang, who's actually about to start a faculty position at Penn State. And Yifei's idea was to throw out the discrete clustering completely from the FitCons method, and instead to define a linear model. In general, it's a neural network, but we end up using a network without hidden layers because it works just as well as one with hidden layers. And so it ends up being a linear logistic model. And we just consider at any position in the genome a vector of features of all possible features that might be relevant to fitness. So we end up using around 50 different features. Some of them are epigenomic, some of them are annotations. We even throw the evolutionary conservation scores in there. And then those features feed through a network. We take a linear combination of those features, feed it through a sigmoid function, and they define the parameter rho, as well as another nuisance parameter of the model. And then we can compute the likelihood of the data using the probabilistic model that I just described for you. So this is kind of interesting. It's a generative model, but it's conditioned on a generalized feature vector at every site in the genome using a linear logistic model, or more generally a neural network. And we can fit the parameters. The free parameters of this model are now the weights of the edges in this neural network. We can fit those parameters by maximizing the likelihood of the data. So even though this looks kind of like a neural network for classification, there's no classification here. This is unsupervised learning. It's being fit to the data purely by the evolutionary patterns in the data, and we're learning which features are most predictive of evolutionary pressure on this time scale of human populations and human-chimp divergence. And this method turns out to work extremely well for predicting disease-associated non-coding genomes. I'm the host of the meeting. That's nice. And we've also used it for, get rid of this. And we've also used it for prediction of active regulatory elements from data sets not used as features. And we find that it has good predictive power to identify regulatory elements, microRNAs, and other types of interesting functional elements across the genome. The second method that I want to mention very briefly, I think I lost my. The second method that I want to mention very briefly is sort of an extension of this LINSIGHT model. We call this LASSIE for linear allele-specific selection inference. And the idea here is to generalize the previous method in a couple of different ways. First of all, we don't consider just the probability that a mutation of any type will have fitness consequences. We differentiate between all possible mutations at every site. So that's why we use the term allele-specific. So for example, at a particular site, we now may make a different prediction for a mutation to an A and for a mutation to a C. Secondly, we're able, using a population genomic model, to make actual predictions of selection coefficients. So instead of just a generalized probability that a mutation will have some effect on fitness, we're now able to get out an actual meaningful selection coefficient for every possible mutation at every site. And this is done by computing the likelihood, not using the probabilistic model I showed you a few minutes ago, but using a Poisson random field model based on diffusion theory approximations. And that allows us to parameterize things in terms of actual selection coefficients and to estimate selection coefficients at every site in the genome. So what we end up with is a map like this across the genome, where we can look at every position in the reference genome, think about all possible mutations, and quantify a selection coefficient associated with those mutations. And you can see, for example, that mutations that are synonymous in this case tend to have small absolute values of selection coefficients associated with them. They're nearly neutral, whereas mutations that change the encoded amino acid tend to have much larger absolute values of selection coefficients associated with them. And we also find, I won't show you the results in this talk, but we also find that these turn out to be quite predictive of disease-associated mutations in coding regions. In fact, it performs somewhat better than polyphen and SIFT and some of the other widely used methods for predicting disease association of mutations. Okay, and this work is out in the bioRxiv if you're interested in having a look at it. What I want to talk about today, though, is a slightly different approach to this same problem. So remember I mentioned that the limitation of FitCons is that it doesn't scale well. The clustering step doesn't scale well, and that's fundamentally because the clustering step is agnostic to the underlying evolutionary patterns. So what I'm going to tell you about is a method for clustering, for simultaneously clustering sites in the genome and fitting the evolutionary model. And that's going to allow us to pick out the features for clustering that are most relevant for the evolutionary model, not just the individual features, but the combinations of features. All right, but in order to do this, we need some sort of an objective function. And so the objective function we started thinking about is a measure of entropy. And as we go forward, I'll explain to you some properties of that entropy measure and why it works well in this setting. So imagine that we have a section of the reference genome, and we have some clustering scheme represented by these colors. So here I've got 3 clusters represented by red, yellow, and blue, and I want to compute the entropy of this clustering scheme. Well, the measure we started out using, I'll show you in a few minutes that we end up using something slightly different, but the measure we started using was just the Shannon entropy of the point-wise row scores. So for a binary random variable, Bernoulli random variable that is the outcome of a coin flip, for example, the Shannon entropy is just the probability, in this case that's going to be rho, times log rho, plus 1 minus rho times log 1 minus rho, just simple Shannon entropy. What we're going to do here is we're going to sum that value across all of the sites in the genome, and we're just going to use the row associated with the cluster at that site. So this FitCons method that I told you about has the property that there's a single estimate of rho for every cluster of sites. So the notation here is that the row Cj means the row associated with the cluster that site J is in. Okay, I apologize for the double subscripts, but it makes it compact to describe the overall entropy in these terms. And of course, since there's only one value of rho for every cluster, rather than summing over all sites, I can simply sum over all of the clusters, and I can weight the entropy by the number of sites in that cluster. Okay? So what does this give us? I should mention this is work by Brad Gulko, a student who recently finished up in the lab. What does this give us? Well, it's gonna, if we optimize this entropy, if we minimize this entropy, it's going to tend to find clustering schemes that push rho towards 0 or 1. Why is that? Well, it's because the entropy is minimized when of a Bernoulli variable is minimized when the probability is close to 0 or 1, and it's maximized when the probability is close to 0.5. In addition, it's going to tend to find clusterings that separate the colors by row. It's going to try to find clusterings that give you quite different rows. In fact, you can prove this mathematically using Jensen's theorem, that if you have a cluster of sites that is actually a mixture of two row values, then you have to minimize the entropy by separating it into two clusters that have those two separate row values associated with them. Okay, so this is going to have two desirable properties: push us towards 0 and 1, and make the row values distinct. Both of those are desirable from the point of view of finding a good clustering across the genome. Now, how are we going to do that? If you're computer scientists, you might recognize that there are going to be exponentially many possible clusterings across the genome, and there's no good way of coming up with a dynamic programming algorithm or something for separating them. So it's going to be a computationally intractable problem to find an optimal clustering, but we can do something pretty good by recursively splitting one cluster into two clusters and picking the split that gives us the largest reduction in entropy. So what does that mean? Well, if we have a collection of 3 clusters, let's make it concrete. Let's put a covariate next to them. So one cluster might represent low RNA-seq, another one, or no RNA-seq signal, another one low RNA-seq signal, and another one high RNA-seq signal. At an intermediate step in this recursive algorithm, that clustering would be captured by a tree. So for example, our tree might first split the no RNA-seq signal regions from the some RNA-seq signal regions, and then later split the low from the high RNA-seq regions. And now, when we consider a new covariate, such as DNA-seq, we're going to find a bipartitioning of the sites that most reduces the entropy of the overall clustering. So in this case, we would consider 3 possible ways of splitting things. We could either take the blue sites, the no sites, and split them into the ones that have low DNA-seq and the ones that have high DNA-seq, or we could do the same for the low RNA-seq sites or the same for the high RNA-seq sites. And what we'll do is we'll find the one that leads to the most reduction in entropy, the most informative clustering from an evolutionary point of view, and choose that as our way of further partitioning the sites. And by doing this recursively, we can grow a decision tree that's going to find combinations of covariates that are informative about natural selection, okay? And we'll effectively be simultaneously fitting the evolutionary model and finding the clustering. We'll be doing it using a greedy algorithm that's not guaranteed to find a global optimum of the entropy function, but it will be guaranteed to find a local optimum. We'll find a locally optimal solution to this optimization problem. And yeah, and as I mentioned, the clustering scheme will be guided by both the epigenomic data and natural selection. And as I mentioned earlier, the clusters will tend to be homogeneous and distinct from one another. Okay. Now, there's an interesting byproduct that comes from this type of clustering, and that's that as we pick new covariates and use them to further split partitions, we will necessarily reduce the entropy. That's by construction, right? We're making choices of bipartitions that reduce the entropy. But this reduction in entropy is interpretable. We can think of it as the information associated with a partition in the genome. And if we sum that reduction in entropy across all partitions associated with a type of feature, let's say DNA-seq data, then this gives us a meaningful measure of how informative DNA-seq data is about natural selection, right? That's exactly what we're measuring here. We have a measure of entropy in terms of natural selection. We're finding ways of using annotations or epigenomic data sets to reduce entropy, And the reduction in entropy that comes from the use of a particular feature is a measure of how informative that feature is. So this gives us an interesting way of thinking about the question, how much do we learn from the genome? How much do we learn about the genome by using a particular epigenomic data set? DNA-seq data, ChIP-seq data, histone modifications, whatever. It gives us a way of putting them all on the same scale and comparing how much we learn from each of these different types of measures about evolution and about genomic function. Okay. Right. So why is this relevant? Well, there's a lot of controversy in the field about how informative these various epigenomic measures are. How much do we actually learn? How much of the genome actually is functional? How much of it is just being picked up by these epigenomic data sets incidentally because of incidental protein DNA binding or incidental chromatin accessibility? And this framework potentially gives us a way to study how much you actually learn from an evolutionary point of view about the genome by using these epigenomic data sets. Okay. All right. Now, I'm running out of time, so I'm going to jump into our results in a second here, but I just want to mention briefly that what we actually use as our measure of information is the likelihood, the log likelihood function from the INSIGHT model, rather than this measure of entropy based on the binary measure, the binary row values. This turns out to be closely related, but richer measure of entropy and gives us some advantages. And I'll refer you to our paper on this topic for details. What that means is we can approximately minimize the entropy by maximizing the log likelihood, and the reductions in entropy accomplished by using a new feature can be estimated by increases in log likelihood that result from including a feature in the analysis. Okay, so this is how the method works. We start with a bunch of epigenomic data sets, RNA-seq, DNA-seq, Chrome-HMM, whole genome bisulfite sequencing, and others. We also throw in a bunch of annotations. Now, we use the epigenomic features separately for each of 115 cell types studied by the Roadmap project. We're actually going to fit the model jointly to all 115 cell types by averaging over them. So this is a fairly big data analysis project. And then on every step of the algorithm, we look at possible binary splits associated with each of these features. We consider many possible splits, and we choose the one that is most informative, and we also choose the point on the tree where it's most informative. And in this way, we grow a decision tree that allows us to classify sites in the genome. And this is what we end up with. We end up with when we analyze the Roadmap data, we end up with this very rich, quite complex decision tree. I won't go through all the details, but I'll highlight a few things. The first thing it picks is a split between coding and non-coding regions, not surprisingly. Secondly, it starts to use Chrome-HMM features, uses them in different ways in coding and non-coding regions. In coding regions, it picks out the Chrome-HMM features that are most associated with active regulation of transcription. Those tend to be close to exons. It then picks out the sites that are within splice sites. That's a small but very high information subset of sites close to exons. In coding regions, it distinguishes between regions that are more highly transcribed and regions that are more lowly transcribed. Later on, it takes advantage of RNA-seq data, and it also makes a distinction here between synonymous sites and non-synonymous sites, roughly speaking. The measure we use is slightly different, but it's essentially picking out the synonymous and the non-synonymous sites. And then in non-coding regions, it uses other combinations of features. And then we can map these scores back to the genome and get a measure across the genome of individual FitCon scores. We can do this in a cell type specific fashion. And these features are quite rich. So you see the codon structure inside coding regions. You see that they're picking out individual transcription factor binding sites in promoter regions, and you see the splice sites showing up very nicely, and a number of other fairly low-level features show up in these scoring schemes. All right, I'm out of time, but I'm very briefly just going to mention a few high-level findings. First, the overall amount of information, not surprisingly, is dominated by coding sequences, but we also get quite a lot of information from RNA-seq and Chrome-HMM, as well as other features. And we can look at combinations of features that are particularly important. We call this synergy. We can measure synergy as the reduction in entropy that comes from considering two features in combination minus the reduction that comes from considering each of them individually. And interestingly, when we do this, we find that many of the epigenomic features are synergistic with one another, and in particular, the DNA-seq feature is highly synergistic. And we think the reason is that DNA-seq is the single best indicator of whether regions in the genome are actively are active or not, are actively transcribed or actively regulated. And so it turns out to be particularly informative in combination with both annotations like transcription factor binding sites, because binding sites that are active are going to show a stronger signature than ones that aren't active, and with other epigenomic marks. We also see quite a lot of synergy from Chrome-HMM. All right, I'm going to skip this part. This is an attempt to partition contributions to entropy from selection and neutral processes such as mutation and drift. And I'll refer you to our paper, which is in press at Nature Genetics for this analysis. And I'll also skip this slide, which shows that the cell type specific scores, if you cluster on the basis of those scores, they recapitulate the known relationships among cell types in the roadmap data set. All right, so what have I shown you? I've introduced this method for measuring natural selection on recent timescales across dispersed collections of elements based on patterns of polymorphism and divergence. I showed how that framework can be sort of hijacked to measure fitness consequences of mutations. And then I showed you a bipartitioning algorithm that lets us simultaneously find a good clustering and estimate the parameters of an evolutionary model, which we've done for 115 different cell types from the roadmap project and made available maps, FitCons maps, for those cell types specific FitCons maps. And then I showed you how, kind of interestingly, this measure, these reductions in entropy, can give us an idea of how much information, in an information theoretic sense, is associated with different parts of the genome. And we find, among other things, some evidence of synergy between data types, particularly with DNA-seq, and then some other findings that I didn't have time to go into. So I'll stop there. I want to thank Brad Gulko, who was the lead on the FitCons 2 project that I talked about at the end, Yifei, Ilan, and Leo, who led earlier aspects of this work, and a number of different funding sources that have helped make this project possible over a course now of, I think, almost 8 years. We've been working on aspects of this project. And then, and finally, I'll just thank my whole lab group who've been a pleasure to work with. Okay, thank you very much, and there might be time for 1 or 2 questions.
Is this working? Okay.
Yeah.
Yeah, I have a question regarding your algorithm with the kind of the bipartitioning. I guess the underlying principle there was like row has to do with selection, right?
Yes.
Have you considered using the same algorithm but using a different metric for row, like something like GWAS association statistic or something?
Right. That's a very good point. We've talked about that, but we've never actually done it. But you make a very good point that the system is completely decoupled, really. The algorithm for forming the tree is decoupled from the objective function. So you could, in principle, use a completely different objective function, but we have not done that yet.
Yeah, that'd be cool.
Yeah.
Yep, here.
Yep.
So thank you for this very interesting talk. So you have shown that your computations are based on the summing above 100 cell types, right?
Yeah.
So have you tested how robust is your score to the amount of information, so the amount of cell lines that you are including?
Yes.
Okay, so first this one.
Yes, we did, in response to a reviewer, maybe it was you, we did a subsetting analysis where we subsampled collections of cell types, and we found that it was quite robust to the cell types. Now, it's not particularly robust to the choice of the individual features, because meaning the individual epigenomic marks that are used.
Okay, that was my second question.
Yes, because the formation of the greedy formation of that tree can go in very different ways depending on the epigenomic data sets that you have present for any given cell type. The qualitative results are similar.
Yes.
Similar regions of the genome will have high or low scores, but the details will end up depending quite a lot on the choice of epigenomic marks.
So one of the interesting results is this synergy conclusion, right?
Mm-hmm.
So like a DNA-seq is more synergistic with other type of epigenomic markers. But do you have also some kind of insights of which of those are more volatile for the amount of data that you include? So that's, I mean, the conclusions about the relevance for the model change more depending on how much data you put in the model?
Yeah. I mean, we didn't do an analysis of that type for the paper. My intuition is that the synergy result will hold, I think, as long as you have something in the model that captures activity. The nice thing about DNA-seq is it's one of the reasons people use ATAC-seq or DNA-seq so heavily is that it's a relatively simple but effective way of describing which regions of the genome have open chromatin and are doing something in a particular cell type. So I think if you had another feature that got at some of those properties, like ATAC-seq rather than DNA-seq, and to some extent Chrome-HMM picks up on the same things because some of the histone modifications also are reasonably good indicators for activity. As long as you have something in the model that picks up on that property, I think you will see something like that synergy. Yeah.
Thank you.
Okay.
Adam, as you well know, the word function and thank you. Okay. The word function and fitness and all of these are very loaded words. So,
yeah.
In what level are the estimates that you get that of really fitness? I can see, I understand it in the FastCon sense, but
yeah.
In your recent epigenetic work or using these data, they are an index of non-randomness. I give you this. But in what sense are they really fitness differences? I mean, what's the data that you bring in to say that will lead to, you know, the usual kinds of
that's a very good question, and it is important to be precise here. So, so these estimates, there's several layers to this question. So I'm glad you asked it. So these methods based on INSIGHT are measuring the probability that a point mutation will sufficiently impact fitness to change allele frequencies beyond what would be expected under neutrality. That's essentially what they're doing. Now, they're point mutations only, right? So that's an important caveat here. So there are many types of mutations that might have many different effects on fitness. Duplications, insertions, and deletions. Those types of mutations, by changing the spacing between elements, could change fitness in a way that's not detected by this method. We're really measuring point mutation effects on fitness on a particular time scale, on the time scale of human polymorphism and divergence with respect to chimps in this setting. Now, I showed you that other method that actually lets us get at this method, LASSIE, that actually lets us get at true selection coefficients. That's a slightly different measure from what we're doing with INSIGHT, which is giving you a value between 0 and 1 from a mixture model that it's basically based on a mixture decomposition. It's saying you have to have some impact of mutations on fitness at some fraction rho of a collection of sites in order to explain the genome-wide patterns of polymorphism and divergence. Now, the third layer, I'll finish in a second, I know you're trying to move on. The third layer has to do with the cell type specificity, and that's a strange feature of this model that we've struggled with for a long time. So the model gives you a different measure of fitness for every cell type. But of course, fitness is not really defined at the cell type level, it's defined at the organism level, right? So what it's really saying, if you get a different measure of fitness at a particular nucleotide in, liver cells and blood cells, what it's really saying is that site in the genome has marks in liver cells that are associated with such and such level of fitness. And in blood cells, that mark is associated with another collection of sites that have a different measure of fitness, have a different effect on fitness. So it's not really cell type specific fitness, it's a reflection of cell type specific epigenomic fingerprints that group sites differently in different sites. We can talk more about it offline.